Good afternoon, everybody, and welcome to EFSA's webinar. We are very excited today to present to you our draft opinion on protein safety assessment in GMOs. This opinion is currently under public consultation. My name is Renhilde Schoonjans, and I'm the team leader of the GMO Food and Feed Safety Assessment team. I will be the moderator of today. Thank you for joining us so numerously and showing great interest in our work. We have received many registrations representing diverse stakeholder groups, which include food industry, universities, NGOs, and competent authorities from Europe and also beyond. So some of you may be following us in the early morning or the late evening, and we thank you very much for participating. You are automatically connected in a listening mode only. This event will be recorded and it will be published together with the presentations on EFSA's website. Throughout the event, which will be in English, you can submit questions. Please do that in the Q&A chat function on the right side of the on top of the presentation window. Your questions will be checked by our Q&A managers and passed to the speakers. We want to ask you, please stay in scope of this webinar, which is to clarify the key components of our opinion and the rationales behind. We can also accommodate questions to clarify any doubt regarding the public consultation phase. Out of scope of this webinar, however, are clarifications on legislations or risk management aspects related to the authorization of a given product. So we would also like to please keep your questions short, since we will address as much as we can during the live Q&A session at the end of the presentations. As you can see here on this slide, we have EFSA team of colleagues here with me to provide the webinar, and the two expert presenters among us are Antonio Fernandez, who is Scientific Officer in the Nutrition and Food Innovation Unit, and Javier Moreno. He is the chair of the GMO Food and Feed Working Group. As Q&A managers, we have Telemakos Gumperis, Michele Ardizzone, and Elena Sanchez-Bronete. So again, they will be handling your questions that you submit through the webinar via the Q&A chat. This webinar is a follow-up to our previous webinar that was held in December 2023. The structure of this webinar will be that Antonio is going to introduce to you in his presentation of about 15 minutes the reasons and the objectives of our draft opinion on protein safety assessment in GMOs. This presentation will include also the ways that we have started to incorporate this approach into our work and in some dossiers. Javier will then explain in more detail the key elements of the opinion and also the next steps that we have envisaged in our recommendations section. Our expectation is that during the public consultation, you will share with us your best practices and gained insights for such protein risk assessment. So during our live Q&A, then at the end, which will last for about 20 minutes, we will respond to your posted questions. So why are we having a public consultation? This public consultation is open for everybody, and we adhere to transparency and openness to ensure that EFSA scientific work and opinions are clear and open to external scrutiny, so that our work is grounded in robust scientific evidence. Also, stakeholders' engagement is important to us to foster diverse perspectives and promote inclusivity in EFSA's work. The comments from this public consultation will allow us to finalize the draft opinion as accurate as possible with enhanced quality and relevance. So please Scan this QR code and you can explore how to submit comments in this electronic tool. This tool will remain open until the 12th of March 2025 to allow as many responses as possible from all stakeholders. And with this, I would like to pass the floor to Antonio Fernandez, Scientific Officer of the NIF unit. Thank you.
Thank you, Renilda. And welcome to everyone. Good morning, good afternoon. Thank you for joining today to this webinar, where, as Renilda was presenting, we are going to introduce to you the draft scientific opinion on protein safety in GMOs. Before entering into the document, I will provide to you a short background information to explain the reasoning behind the document. So, by protein safety today, we refer specifically to protein toxicity and allergenicity. And in terms of risk assessment perspective, it's important to mention that Codex back in 2003 defined the principles for protein safety that are internationally recognized. This was 20 years ago, and specifically for Europe, EFSA and EU regulation has borrowed such principles, and the main elements included in our requirements for protein safety assessment include information on knowledge on the protein, on the source of the transgene or the protein itself, where specifically history of safe use is mentioned, bioinformatic analysis, in vitro studies, and in vivo studies. I will come back to this later on in the next slide. With this context in mind, the EFSA GMO panel has gained experience over the last 20 years and highlighted the need to work further on three main elements: the need for criteria on specific aspects, reflect on the challenges on the assessment of the newly expressed proteins, and to introduce, taking on board new methodologies. All these three aspects interconnected and, as previously mentioned before, with a clear international context. In particular, just to give you some examples in relation to criteria, we have discussed in the document the need to pursue further what is meant with similarity, how similar is similar, how to define history of safe use or how to use it in a broader context as what is done at the moment, and how to move, if possible, with additional thresholds of what we have at the moment. In terms of newly expressed proteins, the GMO panel has taken experience on difficult cases for proteins that are difficult to extract and purify. So how to move forward? Is it developing purification, additional alternative purification systems or alternative expression systems? GM plants with a high number of proteins, creating a whole bunch of challenges to overall safety assessment, and whether or not there is an agreement on testing the mixture or test each individual proteins as we do at the moment. Those cases of proteins with partial similarities to toxins and allergens and the need to develop more targeted in silico tools or hypothesis-driven in vitro tests. In relation to methodology, as you will see later, the need for using these complementary alternative methods in the current approaches, the need to have experimental shift within the weight of evidence approach, and last but not least, always with the international context in mind, the need to standardize the methods used. Next, please. For the preparation of the opinion, the GMO panel also consider up-to-date knowledge, and the EFSA has commissioned work to external experts via developmental projects. They were published in the last year, and they involve articles on allergenicity assessment, developing novel approaches to increase the reliability of predictions, toxicity assessment exploring in silico and in vitro tools, effects of processing with its relevance on protein safety, the development of an adverse outcome pathway on celiac disease, and a work we are doing together with OECD. We also published a software on peptide binding prediction for celiac disease as well, and has worked on methodology for open reading frames. Having all this context in mind and now entering into the document itself, the GMO panel has been mandated to develop a scientific opinion reflecting on current practice, challenges, and future opportunities in the protein safety of GMOs. And the document has been structured in 4 sections that are coming from the mandate as terms of reference. Terms of reference 1, lessons learned from experience in the assessment of newly expressed proteins in the last 20 years, including more recent complex cases. Terms of reference 2, building upon the experience and issues identified, develop a critical appraisal of new methodologies, as mentioned before, that can be used as complementary or alternative to what we are doing now. Terms of reference 3, roadmap for future implementation of these complementary alternative methods. And last but not least, recommendations for further development and research to address methodological needs. Having in mind as a goal, more effective assessments, using less number of animals, speed in risk assessment, and all emphasizing stakeholder engagement. Entering into the terms of reference 1, we will go through them, each of them now. Starting with the problem formulation, the lessons learned, we explain the current approach for protein safety, highlighting the overarching information currently considered that includes molecular and biochemical characterization, bioinformatics, in vitro analysis, in vivo, and most importantly, reflecting in the opinion on the challenges, the aspects requiring further considerations, and the need to make use of novel strategies, providing specific examples to better represent the issue. Just to give you some examples, the need for a nice manner to address substrate specificity, we included there specific examples in that direction. The fact that at the moment, bioinformatic analysis currently ongoing using primary amino acid sequences, the need for fit-for-purpose databases, the fact that at the moment we only use in the assessment the pepsin test that only consider gastric digestion, history of safe use, for which we don't have consensus at the moment. And furthermore, it was also found difficult, this is what is mentioned in terms of reference 1, how to apply these requirements into more complex cases. Next, please. Yeah, exemplifying proteins that are difficult to produce and isolate, some of them difficult even to just have a protein characterization. Proteins with potential hits to toxins and allergens, so how to move further in the assessment. Most importantly, difficult cases of GM plants with numerous proteins to assess. And last but not least, also for the future, proteins that are new to nature. In terms of reference 2, we develop a new methodology with a potential to be used as complementary and alternative methods, starting with the in silico analysis for allergenicity, moving towards the ranking of proteins with different allergenic potential. In terms of toxicity, to move towards machine learning methods and highlighting the importance of datasets used in this assessment. Functional and structural similarity, highlighting the 3D structure comparison and modeling, how to define best practice. Phylogeny, bringing together evolutionary and structural characteristics of proteins into the assessment, and protein aggregation, providing some examples of programs from artificial intelligence that are promising for prediction. In terms of in vitro studies, protein characterization, progress is made with mass spectrometry. Enzyme substrate specificity, examples included on better use of in silico and in vitro methods for more robust assessment. Protein degradation, novel protocols that are currently used also for the scientific field, and what criteria can be used to identify relevant fragments after the digestion. Protein toxicity to be performed in a hypothesis-driven approach. And in a second step, in vivo studies, always also in the terms of protein toxicity, to be used on a case-by-case basis, and when needed, to develop targeted studies that are focused on the safety assessment of proteins, protein allergenicity, protocols that merge food allergy and toxicological assessment. Last but not least, in this terms of reference 2, where we explain the potential of different complementary alternative methods, we're reflecting on exposure and how the role of it can be used further in the safety assessment, also when linked to thresholds, referent doses, and etc. So to illustrate how this information that we included in terms of reference 2 could be integrated in the weight of evidence approach, we built a figure that with case studies that will be presented to you by Javier. So with this, I stop. I thank you all, and Javier, please.
Okay, thank you, Antonio. Hi, everyone. Just let's move on the term of reference 3, which provides key aspects on how to implement complementary and alternative methods in risk assessment strategies. The proposed roadmap reinforces the weight of evidence approach in a structured, stepwise manner, prioritizing in silico and in vitro methodologies while reserving animal studies for cases where a hypothesis needs testing or critical safety data is missing. The figure you can see in this slide illustrates this tiered proportionate framework, which provides flexibility, allows for case-by-case evaluation, and helps establish clear criteria for decision-making in protein safety assessment. Now, let's dive deeper into the key components of this figure. In the next few minutes, we'll break down each of the different stages. So with that, let's move on to the next slide, please. Yeah, thank you. The process begins by evaluating whether the safety of the newly expressed protein or its donor organisms is described in scientific literature, is the step number 1. If evidence supporting safety exceeds, we proceed to the next step, number 2. If not, alternative evaluation strategies are required, and they will be explained later on. If evidence is present, we move to the left of the figure to evaluate whether the newly expressed protein or donor organism has a history of safe use. A well-established history of safe use allows us to proceed to the next criterion, number 3, which is the sequence identity between the newly expressed protein and non-safe proteins. A 100% sequence identity obviously strongly supports safety, requiring minimal further protein characterization. If the identity is lower, additional evaluation strategies come into play depending on the degree of similarity. These may include biochemical or functional assays of the newly expressed protein, structural analysis, or comparison with toxins and allergens. When the history of safe use is unclear, the assessment incorporates additional factors like protein familiarity and phylogenetic relationships. By analyzing structural similarities with known proteins, we can potentially predict a safety profile without needing specific in vitro or in vivo studies, as long as sufficient information is available. Essentially, if a protein is closely related to a well-established safe protein, its familiarity could justify waiving certain experimental requirements. Next, please. Yes, going back to the top of the figure to the right of the slide, when no data is available on the safety of the newly expressed protein or its donor organisms, additional analysis may be required. These include in vitro studies to assess protein stability, which may involve specialized protocols that simulate the gastrointestinal environment, allowing the study of the protein behavior under conditions that closely resemble the human physiology. Additionally, other studies may be conducted to evaluate the effects of pH and temperature on protein stability. If these assessments reveal no hazards and provide sufficient information, further testing may not be necessary. However, when hazards are identified, additional in vitro or in vivo testing becomes important. If no hazards are found but critical safety data is missing, the approach allows for tailored risk characterization considering intended uses and exposure levels. In summary, this approach is designed to be efficient, ethical, and scientifically robust. It ensures that we collect the important data while avoiding unnecessary testing. If you have the opportunity to have a look to the scientific opinion, this approach has been tested with both real and hypothetical case studies, showing its reliability and adaptability across proteins with diverse structures and functions. Next slide, please. Yes, upgrading in silico, we consider that upgrading in silico tools is a crucial step in enhancing protein safety assessment. These computer-based models may simulate protein behavior under different environments and assess risk by comparing structural and functional similarities to known toxins, allergens, or safe proteins. Enhanced in silico modeling could increase the accuracy of prediction, helping to identify potential issues earlier in the process, in the risk assessment process. In this slide, we try to summarize and explore how modern bioinformatics and artificial intelligence-driven tools may improve the efficiency, accuracy, and reliability of safety assessment compared to traditional methods. For allogenicity prediction, conventional approaches, as you probably know, rely on sequence identity threshold, which in some cases could lead to false positive. Alternative models, by incorporating structural features and epitope mapping if they are available, may reduce this false positive, potentially improving prediction accuracy and minimizing follow-up testing. In a similar way, in toxicity predictions, traditional methods depend on sequence similarity to known toxins. But there are advanced models that could improve, again, accuracy by considering toxin mechanisms, mode of action, and functional motives, moving beyond the simple sequence alignment. Also, 3D structural analysis has also evolved, as you probably know, while traditional methods focus on basic sequence alignment. New, robust, and scientifically sound computational tools could predict three-dimensional structures and allow for more comprehensive functional assessment, potentially improving our understanding of protein function and behavior. In phylogenetic analysis, artificial intelligence-driven evolutionary clustering algorithms may provide a more reliable approach for identifying functionally similar proteins, helping us to determine safe evolutionary families with greater confidence. And the scientific opinion contains some of these practical examples. Finally, protein aggregation risk, which traditionally require expensive and time-consuming experimental studies, could now be predicted using advanced models detecting aggregation potential before in vitro testing is even needed. By integrating these advanced methods, we may achieve faster, more accurate safety evaluations, reducing reliance on animal testing. Next slide, please. In this slide, we have summarized key aspects of terms of reference 4, which focuses on identifying gaps and uncertainties in the current protein safety assessment process and the research and developmental activities needed to address these gaps. Clear criteria are needed on how to incorporate history of safe use and protein familiarity into risk assessment, ensuring a consistent and appropriate application. Establishing criteria for best practices and validation of in silico tools, along with selecting suitable comparators, is essential to improve the reliability of predictions based on new in silico methods. The development of relevant in vitro protocols with clear criteria for interpreting results is crucial to enhance their applicability in protein safety evaluations. More targeted studies are required to focus on potential hazards associated with proteins, improving the specificity and relevance of in vivo testing. Next, please. Yes. Exposure considerations need also to be more effectively integrated into the overall safety assessment of newly expressed protein, ensuring a comprehensive evaluation of potential risk. Developing surveillance systems to confirm safety assumptions and reduce uncertainties as proteins enter in the market is also critical. For instance, predicting de novo allergenic potential remains a significant challenge, and more research is needed to develop reliable predictive models. For complex cases as the ones Antonio presented before, alternative risk assessment strategies should be explored to ensure safety is appropriately assessed. And finally, additional factors such as the impact of processing, food matrix, and omics technologies could be integrated into the safety assessment process in the future. Next, please. Yes, so EFSA welcomes feedback on the necessary developmental and research activities and seeks input on how this should be prioritized. This feedback will help us to guide future research efforts and refine methodologies to improve the safety assessment of protein. So now we are already in the last slide. Now let's review the main conclusions and key messages contained in the draft scientific opinion. The main purpose of this document is to critically evaluate the overarching framework for protein safety assessment of newly expressed proteins. This document aims to highlight key challenges and gaps within the current safety assessment processes and explores potential solutions to address these issues in the near to medium term. However, it's also important to note that this document is not intended to provide guidance on the current assessment of the newly expressed protein. Also, as scientific knowledge and technology continue to evolve, risk assessment strategies must also adapt. So there is a need for discussion and consensus on how new methodologies should be integrated into the weight of evidence approach for newly expressed proteins and on their value when combined with current testing protocols. Next, please. Yes. So the EFSA GMO panel remains committed to reducing or replacing animal studies while ensuring high levels of consumer protection. A stepwise approach is recommended where animal studies or more targeted in vitro testing are only employed when strictly necessary. Finally, it is a timely opportunity to revisit and clarify the purpose of protein safety risk assessment, ensuring that newly expressed protein in GMOs are evaluated in a manner that is appropriate, consistent, and proportional. Next, please. Yes, this is just to remind you that Renilde said at the beginning of this webinar that the open consultation period is available until the 12th of March. So your contributions are key in shaping a robust and scientifically sound framework for the safety assessment of newly expressed proteins. So next slide, please. So following the public consultation process, the document will undergo a revision based on the comments received. The adoption of the updated version is planned for mid-2025. However, we hope that this will not be the end of the story, but rather a living document with a longer journey. So some of the foreseen activities are highlighted in this slide. These include a targeted online event prior to adoption, also a webinar to present the final documents once published. Time will be allocated for all stakeholders to share experiences and further build on the collective knowledge. And work will continue towards the development of criteria and reaching consensus on key elements for the ongoing assessment process. These activities aim to foster collaboration, enhance the document relevance, and ensure its continued evolution in line with emerging scientific advancements. So we look forward to ongoing collaboration and further development in this area. So thank you very much for your attention. And now I give the floor to Reinilde. Thank you.
Yes, thank you, Javier. We have reached a point in our webinar where it's time to address questions. So I would like to encourage everybody to use the Q&A chat. It's a chat function that you find on top of the window where you see our presentations. But thanking a lot the speakers, so detailing why we are looking for new assessment approaches. It comes from real life experience and mostly connected to quite innovative products that we didn't, or that we see coming in only in the last decades, but before that, most of the products were quite in the similar line of mode of actions, but now we are facing a very new type of products. So we are processing a list of questions. The Q&A managers are very busy. We will check them, pass to the speakers. If needed, we will ask you to resubmit them to to EFSA ask function, or even encourage you to submit them directly into the public consultation if that is needed. Before sending a first question to Antonio, Antonio, can you give a most pungent and recent example of this work, from where it originated and how we dealt with it through stakeholder engagement, also refined the way we could have assessed such a product?
Reinilde, I missed the question, sorry.
Before giving you the floor with the question and to answer it, I think it would be interesting to give us a real-life example of a very specific situation that was very difficult to test with the methodologies that we had over the past, and where exactly it was absolutely needed to do something else.
Yeah,
thank you.
Thank you, because I was checking the questions were coming up. Yeah, no, thank you. Yeah, so one of the main reasons why we started with this work indeed was the fact that we are receiving more and more cases where we have difficulties in applying the current requirements. Difficulties, as mentioned, because we might have products containing a large number of proteins. That's a challenge in itself. Are we going to test protein by protein? Are we going to continue to do this in the future, where we know that more and more products might come with 10, 20, 30 newly expressed proteins? That might be unhandleable. That's one aspect. The other aspect that also we discussed was for those proteins that we have even difficulties in characterize them. How can we do the assessment of those proteins from a different angle? Because most of these tests that are coming from Codex were designed for let's say, easy to test proteins. And we are seeing more and more difficulties from that angle. I would say these are the main highlights why we started with all this. And as you said, from the beginning, we have engaged with the stakeholders to make sure that we take on board all the different considerations. We have just now put there our initial thoughts. And the more specific comments we receive from you, please be specific. I would say the better for the outcome of the final document. Thank you. Back to you, Renilda. I don't know if we can start with the questions or.
Yes, please. Yes.
Okay, so I will start with question number 1. Are there any demonstrated examples where similarity only of amino acid sequence was actually an allergen? This indeed is a good question. Under terms of reference 1, you will see that we have included there examples on how the safety assessment is performed. And I would say the key message there is performed in a weight of evidence approach. And we always do it in a stepwise manner. And that means that bioinformatics is to be considered as a first net, I would say. So the fact that a potential hit is identified, it does not mean that it will have eventually biological relevance, but it will mean that further assessment will be needed to understand if that in practice has any biological, is of any biological concern. We have, at least from the experience on applications, very rare cases where some potential hits are identified, but none of them has been considered of biological relevance. And what we have in the opinion reflect further is how we could do this in a more robust way. Also the bioinformatic analysis included there some ideas for future, such as the development of fit-for-purpose databases that can reduce the number of false positives that we can encounter for there, as well as investigating best practices for protein structural analysis, for example. This, I would say, is one of the main highlights that also Javier has been emphasizing on what are the best practices we can develop criteria on for 3D analysis, basically, of proteins. I will give you the floor to you back, Javier. I don't know if you have questions there to continue.
Yes, Antonio, I can take it, some of those questions. Yes, for instance, let me check. Yeah. Okay, Paris. Okay, yes, I have the first one. How does protein stability indicate a hazard? Are proteins like lactoferrin casein considered hazardous by the GMO panel because of it is a rather stable protein? Does stability not say something more on exposure? Okay, this is a good question. I will say that protein stability plays an important role in assessing potential hazard because it can affect how a protein behaves in the body, particularly in terms of the digestion, absorption, and interaction with the biological system, including the immune system. So a protein that is highly stable might persist longer in the gut environment or within the organisms, potentially leading to higher exposure levels or causing unintended biological effects. For example, if a protein is resistant to degradation during digestion, it could trigger immune responses or accumulate in tissues, increasing the likelihood of adverse effects. This is the rationale behind why regulation asks for studying the protein stability studies. But however, the stability of a protein alone obviously doesn't automatically make it hazardous. It's just one of the many factors considering a comprehensive risk assessment. For proteins which are naturally stable, I will say that the GMO panel will assess them on a case-by-case basis, looking at additional aspects such as functional characteristics, the context of expression, and exposure levels. While their stability might suggest that they could persist in the digestive system, it doesn't inherently imply that they are hazardous, as I mentioned before. In fact, these proteins you mentioned, lactoferrin and casein, obviously are part of the human diet, and they have well-established safety profiles. And yes, the question, I think, is correct that the stability can provide insight into exposure, but obviously it doesn't automatically indicate a hazard. So the assessment needs to consider the full range of factors, including stability, potential immunogenicity, and the protein behavior in the specific context in which it's expressed, exposure level, how much of the protein is being consumed, and how it's processed by the organisms are also key elements in understanding potential risk. So I will address another question. Okay, will EFSA guide researchers on how molecular docking could be used as a risk assessment tool? Okay, this is also a very interesting question, comment, as we highlight in the draft scientific opinion. We indicate, we stress that it's important to reach agreement, international consensus about potential bioinformatic thresholds, how this could be established, as Antonio mentioned before, how similar is similar, and how robust are the methods included in this kind of molecular docking. We know that this kind of method could provide good information about how the protein behaves and how similar could be this protein regarding non-safe protein. But for sure, I mean, we need to further discuss this aspect, but the idea is to go into this direction and to reach important agreement about how to interpret and how to include it, this kind of studies in our risk assessment strategies. So I think, Antonio, I will give you the floor for a couple of additional questions. Thank you.
Thank you, Javier. I will continue with a couple of questions. Starting with, we have a good question here. Well, there were a couple of questions in the same direction related to, it is said, multiple slides refer to complementary and all alternative methods. How would it be decided whether a method can be provided as alternative to existing methods? And it's also mentioned by some of you, many new options presented are as additional assessment tools in the expectation that all of these tools be applied on every NEP or should be done on a case-by-case basis. If the former, can you explain how the current framework has not been successful for previous safety assessments? The figure 3 presented by Javier on how the GMO panel considered that new methods can be applied in a complementary or alternative manner has been explained in the document, I would say, with examples. Some of them were real examples, others were hypothetical, to make sure that it is understood that that figure is to be considered on a case-by-case basis. Having said so, I would not consider, or the message we want to give is not that previous assessment has not been successful. On the opposite, the current tools are still valid, are the tools that we are using. The message we are giving here is that we are facing challenges. It does not mean it has been unsuccessful. we are having challenges, and novel technologies can help, or we think can help, to underwent in the direction of having a more robust assessments with the less use of animals, and having a more effective, eventually, risk assessment strategies presented that are more credible. Additional question is related to limitations of in silico protein digestibility and how this might overcome in the future. So in the document, we have a very short section on this, because at the moment we consider is not yet ready to be elaborated. Maybe in the future, as a second step, after gastrointestinal digestion, it could be applied. But for the time being, we could not see any specific recommendation that could be provided on in silico tools for protein digestibility. If, on the contrary, any of you has any examples where we could use that in a different manner, we would really appreciate if you could please include these comments in the public consultation, so that, as mentioned, what we will do once we have your comments, we will discuss again the draft opinion and we'll produce a final version by July. So the more comments, please be specific, we receive, the better. Javier, I pass the floor to you. Yeah, thank you.
Yes, thank you, Antonio. Yes, I will address some more questions. Yes, one of them is related to the post-market monitoring. Could you elaborate on the criteria that might be used to determine what type of post-market monitoring might be required? If a risk safety assessment does not find evidence that an EP will be an allergen or toxin, could post-market monitoring be justified? And how to get, there is a second question, how to get useful data for individual proteins in various aspects of the food supply? Okay, I mean, addressing the first questions, I would say that post-market monitoring plays a crucial role in ensuring the ongoing safety of some neurospace protein after they enter the market. Even if a risk assessment does not identify allergenicity or toxicity, post-market monitoring can still be justified depending on factors like protein type, exposure levels, and the target population. So key criteria for determining post-market monitoring needs include, as I mentioned, protein type, exposure levels, data gaps or uncertainty. If there are uncertainties in the pre-market assessment, post-market monitoring can help fill this gap. Also, unexpected outcomes to detect this kind of unexpected outcome if new health data suggest potential issues. So monitoring could be important. So yes, concerning the second question, how to get useful data for individual proteins in various aspects of the food supply. Okay, yes, I think, for instance, to look for data on comprehensive protein data databases, to have access to information on protein characterization and also to food composition databases, also to monitoring protein levels in the food supply, quantifying protein concentration, also the exposure assessment. Also could be of help to incorporate digestibility data. Or maybe also to use public and regulatory data, and also real-time post-market surveillance studies regarding allergenicity or toxicity monitoring. Also, if there is information available of food processing data to assess the impact of food processing, could also be important, and for sure to have collaboration with food industry and academia. All these activities could be of help to gain data on individual proteins included in the food supply. Okay, I will address yes another question, which is, are transcriptomic data useful for assessing allergenicity, or it should be directly proteomics? I mean, I would say that transcriptomic data can be useful for assessing allergenicity, but in my view, proteomics is generally considered more direct and reliable for evaluating actual allergenic potential. So in that sense, I think it's more robust, the risk assessment, if we have current actual data about the content of the neuroexpressed protein in the product under assessment. So Antonio, I will give you the floor just to address a couple of more questions. Thank you.
Thank you, Javier. Just 3 minutes left to address a couple of more questions. There is one here that says the revision of history of safe use definition is key for risk assessment of GMOs after 25 years of commercialization. It is great to see EFSA acknowledges the gap on this topic. Does EFSA consider revisiting, eliminating the studies currently requested for those proteins that has been assessed numerous times if a revised definition of OSU is to be established? I think this is quite a relevant point, as I think Javier has explained pretty well in the figure 3 is one of the aspects we are moving forward to incorporate not only history of safe use, but also the term we included there. Some scientists has also used familiarity, and how this terminology can be used to move the safety assessment without the repetition of specific studies. I think it's in nobody's interest to repeat the studies, and if a protein has been considered safe or a similar protein safe, the direction we are proposing in that document is to move into this direction. So not repeating studies, but making use of the knowledge as much as possible. It is also mentioned that thresholds has been shown in the past to be easy for the risk assessors, but push the risk assessment into a conservative mode. I think this is a real case. This is a good comment. Would it not be better to use criteria instead of a specific percentage identity criterion to be considered a similar protein? Okay, this goes in the direction of better framing criteria rather than firm numbers, thresholds that might be more easy for risk assessment, which I share this view for many aspects. For others, whenever we have solid grounds, we are also discussing whether we can move into that direction of having thresholds, but most likely it would be impossible. So I guess the development of criteria will be the way to go for the future. With this, I think I will stop the questions. We are reaching the end, so I wonder if we give you the floor back, Renilda. I know there are more questions in there, but just to give you time to finish the webinar. Javier and Renilda, what do you think?
Yes, unfortunately, we are 5 minutes before the end, and these questions are very valuable. They are questions very related to our work. Thank you for this. We will all capture them and think about it, refine the text if needed. But of course, we apologize for not being able to address all of them orally. I am sure they are forming the basis for a good engagement now during the public consultation, maybe after. So thank you so much for this activity in the Q&A chat. I will need to draw this webinar to a closure, and I would like to thank first of all the people here on slide, again, to make it happen. We do prepare carefully to make it meaningful, and we hope you have enjoyed it. So thanks on behalf of all the colleagues you see here. And for your attendance, we do hope you get also something in return from us. If not yet done, in response to your questions, please consider filing them also through our EFSA Ask service, which is a constantly open online tool for whatever questions on products, on legislation, on to find pathways for your product. You are also welcome, of course, again, to join the public consultation by submitting the comments, also those that you gave today. It would be of value to us to see them in the public consultation tool, because we will then have all these comments in an overview file, and together with others, consider them for finalization of our draft, or for further investigation before doing so, or to leave for another piece of work in the future. So please help us by registering there. So the recording again of the presentations will be made available on EFSA's website in the coming days. Give us a little bit of time. We also would like to encourage you to give us back the survey that you will receive by email. I would also like to give a couple of take-home messages from the scientific side. So I saw we are making progress. People asking for criteria, people asking for prioritization in the next steps. And I think an important take-home message is that depending on which type of stakeholder group you belong to, the draft opinion in front of you will give you an overview of what we believe can be done, needs to be done. And we will be very happy to see that being taken forward within the remit of your activities or collaborations to be established. Such priorities can be the history of safe use, how to define it, or novel in silico tools working on the databases to communicate with each other and to be queried, or the potential of novel in vitro testing still to be explored further. So thank you again for attending the webinar. We have a little announcement that depending on the number of comments and the type of comments that we will receive in the public consultation, it may be so that EFSA will consider a follow-up engagement with the stakeholders or the respondents from the public consultation. So this is to be discussed internally, and we could envisage such a follow-up. So looking forward to see you in our next activities and for your contributions to this topic. With this, I close the webinar and wish you all a very good day. Bye-bye.